Hello everyone! In our next experiment, we are going to determine the amount of sulfate in an unknown using a technique called gravimetric analysis. So what are the goals for today's experiment? Well, we are going to determine how much sulfate is in an unknown, and we're going to use a technique called gravimetric analysis, which I'll explain in a minute. And specifically what we're going to do is that we are going to take the sulfate ions that are in our unknown and we're going to convert them to a barium sulfate precipitate, which we can then weigh and then use that as a measure of how much sulfate was present in the unknown initially. What are we going to learn in today's experiment? Well, the skills involve weighing a sample by difference, so we're going to weigh our unknown sample that way. And we want to make sure we can properly set a filtration and quantitatively recover the solids. We also need to understand solubility rules, because understanding the solubility rules allows us to understand why this technique is effective in allowing us to determine how much sulfate is in an unknown. We will also use our knowledge of solubility rules to test to see if a certain procedural step has been completed. So first of all, what is gravimetric analysis? Well, we are going to determine the amount of sulfate that's in our unknown compound, and our unknown compound is an impure salt, and it has sulfate and it has other components in it too, and those components are all water soluble. So if we add barium ions to a solution of this unknown compound, we can form a barium sulfate precipitate. And then we can weigh that precipitate, so that's what the term gravimetric refers to. We can weigh that precipitate and then we can determine how much sulfate was present in the original unknown. We need to point out that this strategy is successful as long as the impurities in the sample also don't react with the sulfate to form an insoluble compound. And that is the case for the unknowns that we're going to provide to you. So what techniques will we learn today? Well, the first one is weighing by difference. So you'll be given a vial of an unknown salt, and you'll weigh the vial, and then you'll transfer what looks to you to be about 0.5 grams of the unknown into a clean 400 milliliter beaker, and then you'll weigh the vial again. And that will tell you how much salt you have added to your beaker. So here's a picture right here of a clean beaker, and here's about a half a gram of the unknown that I transferred to it. We don't weigh the beaker before and after because the beaker weighs quite a lot more than a half a gram, and trying to determine the difference that way would probably yield some inaccuracies. So that's why we weigh the vial instead. So what do we do next? Well, first we dissolve our unknown, and then we add an aqueous solution of barium chloride. So barium chloride is a soluble solution. Our unknown is sulfate, and the manufacturers who supply the unknown to us don't tell us what form the sulfate is in. Sulfate's a negative ion, so it needs to be paired with something positive. I'm going to guess that it's probably sodium, because sodium is soluble and sodium is also inexpensive. So I'm going to guess that our unknowns are probably sodium sulfate. So if we add barium chloride, which is water soluble, to our sodium sulfate, which is also water soluble, we will get a reaction occurs, and this is called either double displacement or metathesis or exchange. There are lots of different names for these types of reactions, but basically what happens is the positive and the negative ions change partners, and we get barium sulfate and sodium chloride as our unknowns, and as our products, sorry. And what drives this reaction is the formation of barium sulfate, which is an insoluble precipitate. So there are many sort of drivers of reactions that go to completion, and formation of a precipitate is one of them. So if we expand this molecular equation to a complete ionic equation, we note that when we take barium chloride and we dissolve it in water, that the ions dissociate to barium ions and chloride ions. The sodium sulfate would dissociate to sodium and sulfate ions. The barium sulfate, though, is insoluble, so it does not dissociate. So we write this as the solid precipitate. The second product is sodium and chloride, and that dissociates. So if we cross out the common ions on each side, the chloride and the sodium ions, then we get the barium ions that we will add will react with the sulfate ions in our unknown to form a solid barium sulfate precipitate. So here we can use molar relationships, and we can see we've got a 1-to-1 mole relationship. For every 1 mole of sulfate that's present in our unknown, we will form 1 mole of barium sulfate. So that knowing how much barium sulfate we form will help us figure out how much sulfate was originally present in our unknown. So when we go into the lab, first what we do is we weigh out our sample like I just showed you, and then we dissolve the sample in a dilute acid solution, and this ensures that all of the components of our unknown are all in solution, whether they're sulfate or something else. Then we add barium chloride, and that precipitates the sulfate ions using the equations that I showed you in the previous slide. So you can see what happens when you add the barium chloride. You get this very cloudy looking solution right here, and the cloudiness is just dispersed particles of the solid barium sulfate. Also you can see that I'm heating this because we want to do some gentle heating to make sure that all of the components of the unknown dissolve in the solution. So we do want to make sure that we precipitate all of the sulfate. So that means we have to make sure that we add enough barium chloride so all of the sulfate precipitates and none of the sulfate remains behind. Here in this is called the supernatant, which is above the solid. So what we do is we test it. We just add a few drops of barium chloride. So I just took some barium chloride and I took a pipette and I kind of dribbled a few drops down the side, and if I see cloudiness when that happens, that's the formation of additional barium sulfate, and that would mean that I hadn't added enough barium chloride. But when I did do that test, I just saw a transparent solution. So that told me that all of the sulfate had precipitated, and I can go on to the next step. But if you do see some cloudiness, you'll have to add another 5 milliliters of barium chloride and then repeat the step just to make sure that you've got all the sulfate precipitated. And when you take it off the hot plate and wait a few minutes, that solid does settle. So now I have a solid barium sulfate precipitate here, and above this is the supernatant, and the supernatant contains sodium chloride ions and other ions that are also present in the unknown sample. So the next step, we heat the solution for about 20 minutes to just below its boiling point. We don't want to boil it because it'll splatter. I did put a watch glass over the top of the beakers just to make sure that wouldn't happen. But the point of doing this is to do something called digestion, and that's a term that's being used here differently. Maybe when you think of digestion, you think of digesting your food, but that's not what this is in this situation. What this means is this: if you gently heat just below the boiling point, then that encourages the formation of very large barium sulfate crystals. Because the next step that we're going to do is we're going to filter it. And the filter paper has small holes that will allow the solution to pass through. But we don't want to have barium sulfate crystals that are so small that they pass through as well. So the digestion process encourages the formation of large barium sulfate crystals that won't pass through the filter paper. It also helps us to form a really nice, orderly barium sulfate crystal, and we don't want to trap any other ions in it either. We want to make sure that we have pure, very large barium sulfate crystals, and the digestion process will make that happen. So while your solution is digesting, you can prepare your filter paper. So as shown in this diagram right here, and it's also described in the video, So you fold the paper in half, and then you fold it in half again, but you don't want the edges to meet. Make sure it's offset by about ½ inch. And then just tear off a little corner, and then unfold it and insert it into a glass funnel with a long stem. And this allows the, if you then just wet it with some water from your wash bottle, you can seat the filter paper into the funnel and provide a nice seal. And if you've done this properly, when you pour a solution through the filter paper seated into the funnel, you'll have a nice steady flow of filtrate through the paper. Because if not, it'll just drip very slowly, and it will take you a long time to complete your experiment. So when the solution is finished digesting, you take it off the hot plate and let the precipitate settle. So here, hopefully, I have my large barium sulfate purified crystals. And then what I do is I put my filter paper in this glass funnel, and then I put the stem of the funnel, so it was, I don't know if you can see this, but I put it right on the edge of this beaker right here, so that when I poured the supernatant into the funnel, I had this nice steady stream of supernatant going through the filter paper. So this should be pretty rapid if you've seated the filter paper properly. So you notice I've poured the entire supernatant here, and I just have my barium sulfate here, and this is my entire supernatant. I had kind of a small beaker here, so at a couple points I had to dump out the liquid and replace it with an empty beaker. Over here I have a hot plate, and you can see that I'm warming up some water, and I did actually measure the temperature to make sure that I heated the water up to about 90 degrees. I didn't want to boil it, but I wanted it to be nice and warm. I also have I don't know if you can see this in my lab bench with all of the other things that I have here, but we have these wooden funnel setups where you could actually do 4 samples at a time. We're going to ask you to do 2, but you could do 4 at a time if you wanted to. So I did 2 samples. So here's my 1 sample right here with my solid barium sulfate. So I was filtering that. I wrote the mass of my unknown on the beaker, but you'll write it in your notebook. So here in both instances, you can see I can do both of these at the same time. I can be pouring my supernatant into my funnel and kind of paying attention to these at the same time. So once all of the supernatant is through the funnel, the next thing you need to do is you need to scrape out all of the solid, and this has to be quantitative, which means we have to get every last bit of it. So we're going to use something called a rubber policeman. It's sort of an oddly named instrument, but it's a glass rod, and on the end of the glass rod is this little sort of rubber piece right here, and it's kind of like a spatula when you're cooking. If you're making cookies, for example, and you want to scrape every little last bit out of the bowl because it's cookies, so of course you would, that helps you quantitatively transfer any of the food out of your bowl. It's the same thing over here. So this rubber policeman will allow me to scrape all of this solid out of this beaker. But it's hard to do that with just the rubber policeman. So you've seen in the background I have a wash bottle. So the next thing I did is I squirted some water into my wash bottle, and I washed the beaker, and I washed the rubber policeman too, to make sure that I get absolutely all the solid out. So that's what we mean by quantitative transfer. We need to get every last bit of it. So I think I've done that. If you look over here, you can see my rubber policeman, and you can see the beaker, and hopefully you don't see any traces of white solid over there. So now my white solid is in my my funnel. But I'm not done yet, because remember, from the initial equation that I showed you, we added barium chloride, and there's probably chloride ions, and not probably, there are definitely chloride ions clinging to that barium sulfate. And remember, we just want the mass of barium sulfate. We don't want any chloride ions clinging to it. So that's where this hot water that I've been heating up, that's where that comes in. And what I did, I think I'll go back to this slide. What I did is I took this hot water, and you have to make sure you handle this. So I used beaker tongs. You could use hot hands, but I used beaker tongs to handle the beaker. And I washed this solid many, many times. I think our manual says 8 to 10 times. I didn't actually count, and you don't have to. And I'll show you why you don't have to. You know when you're finished, what you should do is you should get a series of small test tubes And you can collect some of the drops coming out of the filtrate, and you want to make sure that there's no chloride in it. And the way that you know this is that we have little dropper bottles of silver nitrate. And if I add some silver to the drops that are coming out of my funnel, and if I see a white solid, that's silver nitrate. So you can see with this test tube right here, I still had chloride ions, so I had to continue washing. So what I did is over the course of the washings, I kept testing it, and you can see that these 4 test tubes, that the cloudiness gets less cloudy, and see this tube right here is clear. So as soon as I add silver to the filtrate and I don't see any silver chloride, I know that I have washed all of the chloride off of my solid. So I don't even really need to keep track of how many times I do the washes. I just have to make sure that I wash sufficiently and test it to make sure that I know for sure that all of the chloride is washed off. So that's what my sample looks like. So there's my barium sulfate. So we have drying ovens in the lab, so you'll put it in a drying oven. So you want to dry this for a couple of days. Actually, you want to make sure that this is really, really dry. So so I did this on Friday. So I will come in on Monday and I will weigh the entire contraption of my solid with the filter paper. You would have weighed the filter paper beforehand, so I can determine the mass of the solid by subtracting the mass of the filter paper with the solid from the mass of just the filter paper alone. So when you do this experiment, I had to come to lab dressed appropriately. I had no skin exposed below the waist. My shoes cover the entire foot. I did wear my eye protection and a lab coat and gloves. If you spill any of the solutions on your skin, you should wash them immediately. We're not working with anything particularly dangerous today, but the silver nitrate can stain your skin, so you want to make sure that you are definitely wearing gloves when you handle the silver nitrate. And you want to be sure that you dispose of solids in the appropriately labeled waste containers. So the evaluation for today's work is that in the first 2 experiments we designed tables for you, but what you will want to do in your carbonless notebooks is that you will want to organize the data taken in the lab into tables that you design yourself. And you want to record your data as you go along doing this experiment. And then you will also want to carry out your calculations in your notebook. So today we will do an analytical report. So there is a video posted on Sakai that describes the analytical report, but basically it's just essentially what a results and a calculation and interpretation section would look like. For this experiment, you should be able to design one table that has your raw data collected in the lab and the calculations that you would have done to determine the amount of sulfate in your unknown. And then you'll follow that table with a series of sample calculations. So that will be sufficient for a report for today's experiment. So how do you figure out the amount of sulfate in your unknown? Well, you weighed the filter paper with the barium sulfate precipitate and subtract from that the mass of the filter paper. So you'll know the mass of the barium sulfate, so then you can convert the mass of barium sulfate to moles of barium sulfate. And remember, the barium is what we added, and we just want to know how much sulfate was in our unknown. So we look at the mole relationship, and you can see that there is a 1-to-1 mole relationship. For every 1 mole of sulfate that was present in our unknown, we will form 1 mole of barium sulfate. So once you have the moles of sulfate, then you convert to mass of sulfate, and then when you have the mass of sulfate, then you can compare that to the original mass of your unknown sample, multiply by 100, and you will get the percentage of sulfate in your unknown. You'll do that twice. We asked you to do 2 samples, so you can get an average, but you won't get a standard deviation. And the reason for that is that you need a minimum of 3 samples in order to calculate a standard deviation. So we'll ask you to calculate an average, but not a standard deviation.